Structure of a muscle. Welcome to this video. In this video, we'll explore the structure of a muscle, include what muscle fibers are and how they're organized, the three connective tissue layers, epimysium, perimysium, and endomysium, the internal structure of muscle fibers, including myofibrils and sarcomers, the role of contractile proteins like actin and myosin. Let's dive in and understand how muscles are built to contract and function. Muscle mass, or muscle tissue, is made up of a large number of individual muscle cells, or myocytes. The muscle cells are commonly called muscle fibers because these cells are long and slender in appearance. Skeletal muscle fibers are multinucleated and are arranged parallel to one another with some connective tissue. Muscle mass is separated from the neighboring tissues by a thick fibrous tissue layer known as fascia. Beneath the fascia, muscle is covered by a connective tissue sheath called epimysium. In the muscle fibers are arranged in various groups called bundles or fasciculi. Connective tissue sheath that covers each fasciculus is called perimysium. Each muscle fiber is covered by a connective tissue layer called the endomysium. Each muscle cell or muscle fiber is cylindrical in shape. Average length of the fiber is 3 centimeters. It varies between 1 centimeter and 4 centimeters, depending upon the length of the muscle. The diameter of the muscle fiber varies from 10 microns to 100 microns. Muscle fibers are attached to a tough cord of connective tissue called tendon. Tendon is in turn attached to the bone. Tendon of some muscles is thin, flat, and stretched, but tough. Such type of tendon is called aponeurosis. For example, palmar aponeurosis of palmaris, longest tendon in hand. Each muscle fiber is enclosed by a cell membrane called plasma membrane that lies beneath the endomysium. It is also called sarcolemma. Cytoplasm of the muscle is known as sarcoplasm. And the structures embedded within the sarcoplasm are nuclei, myofibril, Golgi apparatus, mitochondria, ribosomes, and sarcoplasmic reticulum, myofibril. Myofibrils are the fine parallel filaments present in sarcoplasm of the muscle cell. In the cross-section of a muscle fiber, the myofibrils appear like small distinct dots. Within the sarcoplasm, diameter of the myofibril is 0.2 to 2 microns. The length of a myofibril varies between 1 centimeter and 4 centimeters. Microscopic structure of a myofibril. Light microscopic studies show that each myofibril consists of a number of 2 alternating bands, which are also called the sections, segments, or discs. These bands are formed by muscle proteins. The 2 bands are light band or I band, dark band or A band, light band or I band. Light band is called isotropic band, because it is isotropic to polarized light. When polarized light is passed through the muscle fiber at this area, light rays are refracted at the same angle. Dark band, or A band. Dark band is called A, anisotropic band, because it is anisotropic to polarized light. When polarized light is passed through the muscle fiber at 180 degrees this area, the light rays are refracted at different directions. Dark band is also called Q-discy band, is divided into two portions by means of a narrow and dark line called Z line or Z disc. The Z line is formed by a protein disc, which does not permit passage of light. The portion of myofibril in between two Z lines is called sarcomere. Sarcomere. Sarcomere is defined as the structural and functional unit of a skeletal muscle. It is also called the basic contractile unit of the muscle. Each sarcomere extends between two Z-end lines of myofibril. Thus, each myofibril contains many sarcomeres, arranged in series throughout its length. Components. Each myofibril consists of an alternate dark A band and light I band. In the middle of A band, there are a light area called H zone. Electron microscopic study of sarcomere. Electron microscopic studies reveal that the sarcomere consists of many thread-like structures called myofilaments. Myofilaments are of two types: actin filaments, myosin filaments. Actin filaments are thin filaments with a diameter of 20 angstrom and a length of 1 micron. These filaments extend from either side of the Z lines, run across I band, and enter into A band up to H zone. Myosin filaments. Myosin filaments are thick filaments with a diameter of 115 angstrom and a length of 1.5 micron. These filaments are situated in A band. During the contraction of the muscle, the actin filaments glide down between the myosin filaments towards the center of H zone and approach the corresponding actin filaments from the next Z line. Contractile element of the muscle. Myosin filaments are formed by myosin molecules. Actin filaments are formed by three types of proteins called actin, tropomyosin, and troponin. These four proteins together constitute the contractile proteins, or the contractile elements of the muscle. Each myosin molecule has two portions: tail portion, head portion. Tail portion of myosin molecule. It is made up of two heavy chains, which twist around each other in the form of a double helix. Head portion of myosin molecule at one end of the double helix. Both the heavy chains turn away in opposite directions and form the globular head portion. Thus, the head portion has two parts. Two light chains are attached to each part of the head portion of myosin molecule. Each myosin head has two attachment sites. One site is for actin filament, and the other one is for one ATP molecule. Myosin head is absent in the central part of myosin filament. In the H zone, actin molecule. Actin molecules are the major constituents of the thin actin filaments. Each actin molecule is called F-actin, and it is the polymer of a small protein known as G-actin. There are about 300 to 400 actin molecules in each actin filament. The molecular weight of each molecule is 42,000. The actin molecules in the actin filament are also arranged in the form of a double helix. Each F-actin molecule has an active site to which the myosin head is attached. Tropomyosin. About 40 to 60 tropomyosin molecules are situated along the double helix strand of actin filament. Each tropomyosin molecule has the molecular weight of 70,000. In relaxed condition of the muscle, the tropomyosin molecules cover all the active sites of factin molecules. Troponin. It is formed by 3 subunits. Troponin I, which is attached to factin. Troponin C, which is attached to calcium ion. Troponin T, which is attached to tropomyosin. Other proteins of the muscle. In addition to the contractile proteins, the sarcomere contains several other proteins such as actinin, which attaches actin filament to Z line, desmin, which binds Z line with sarcolemma, nebulin, which runs in close association with and parallel to actin filaments, titin, a large protein connecting M line and Z line, sarcotubular system. Sarcotubular system is a system of membranous structures in the form of vesicles and tubules in the sarcoplasm of the muscle fiber. It surrounds the myofibrils embedded in the sarcoplasm. Structures constituting the sarcotubular system. Sarcotubular system is formed mainly by 2 types of structures: T-tubules, L-tubules, or sarcoplasmic reticulum. T-tubules or transverse tubules are narrow tubules formed by the invagination of the sarcolemma. These tubules penetrate all the way from one side of the muscle fiber to another side. That is, these tubules penetrate the muscle cell through and through. Because of their origin from sarcolemma, the T-tubules open to the exterior of the muscle cell. Therefore, the ECF runs through their lumen. L-tubules, or sarcoplasmic reticulum. L-tubules, or longitudinal tubules, are the closed tubules that run in long axis of the muscle fiber, forming sarcoplasmic reticulum. These tubules form a closed tubular system around each myofibril and do not open to exterior like T-tubules. L-tubules correspond to the endoplasmic reticulum of other cells. At regular intervals, throughout the length of the myofibrils, the L-tubules dilate to form a pair of lateral sacs, called terminal cisternae. Each pair of terminal cisternae is in close contact with T-tubule. In human skeletal muscle, the triads are situated at the junction between A band and I band. Calcium ions are stored in L-tubule, and the amount of calcium ions is more in cisternae. Functions of sarcotubular system. Function of T-tubules. T-tubules are responsible for rapid transmission of impulse in the form of action potential from sarcolemma to the myofibrils. When muscle is stimulated, the action potential develops in sarcolemma and spreads through it. Function of L-tubules. L-tubules store a large quantity of calcium ions. When action potential reaches the cisternae of L-tubule, the calcium ions are released into the sarcoplasm. Calcium ions trigger the processes involved in contraction of the muscle. The process by which the calcium ions cause contraction of muscle is called excitation, contraction coupling. Composition of muscle. Skeletal muscle is formed by 75% of water and 25% of solids. Solids are 20% of proteins and 5% of organic substances, other than proteins and inorganic substances. That's a quick look at muscle structure. Like, subscribe, and share with your friends to help them learn. Thanks for watching.